Today in my science class, we're going to look at the fermentation of simple sugars by yeast. That's why I creatively call it the Yeast Fermentation Lab. It's a very simple lab. All we need is some water, some food for the yeast. I've got flour and sugar, and some yeast, which you can buy in the grocery store. I have several different flasks, 5 different flasks, and I'm going to create different conditions for the yeast to grow in. Every flask is going to get water, though, just to provide an environment for the yeast to live in. They're all going to get 100 grams of water. It's from the same source. It's the same type of water, the same temperature, all that. Some of the flasks, 4 out of 5 of them, are going to get yeast. Now, yeast is a unicellular fungus, kind of like a mushroom, but just one cell. It's a heterotroph. It needs to eat other things. In this case, I'm going to feed it sugars. And it performs anaerobic cellular respiration, otherwise known as fermentation, instead of aerobic cellular respiration. It comes coated in kind of a freeze-dried food coating so that it stays fresh. You can just keep it on your shelf until you need it, and then when you dissolve it in water, the yeast will become active. Their coating will dissolve and they'll emerge and start doing their thing. I'm also going to add some food for the yeast to some of these flasks, not all. My 2 foods are sugar, and just to have something to compare it to, I'm going to compare it to flour. Now remember, sugar is a carbohydrate called sucrose. It's basically 1 fructose and 1 glucose molecule stuck together. Its chemical formula is C12H22O11. We call it table sugar, but it's just one of many carbohydrates. You know it's a carbohydrate because it ends in ose, and also because it has that 1 to 2 to 1 ratio of carbon to hydrogen to oxygen. Alright, going to try and be precise with my measurements here. Okay, got my sugar, and I'm going to add it to a few of these. So I'm going to add it to this one here, yeast, sugar, water, and I'm going to boil this one. I'm also going to weigh some out. And I'm going to add it to flask 2. That's just yeast, sugar, and water. And finally, I have this flask that I'm going to get rid of the yeast. I'm just going to have sugar and water in it together. This flask all the way on the right, flask number 5, that's going to be my yeast, flour, and water. So instead of sugar, we're going to try flour. Flour is a more complex carbohydrate, takes a little bit longer to break down, is made of more than just fructose and glucose. There's more to it. Okay, I'm going to take this one that needs to be boiled. I'm going to stick it on my hot plate here. And we're going to fast forward a little bit while it heats up. Okay, it's a few minutes later and it's come to a boil. You can see it bubble. I got to stir it the whole time or else it'll start to cook. But you can see it's nice and boiled there. So I'm going to take it. Let's add it back to the others. Ta-da! Okay, good. And we are ready to begin this experiment. Everything's been added. Lastly, though, we need some way of measuring how much gas is going to come out of these flasks, right? We need some way of measuring the fermentation. So we could measure how much the sugar gets eaten, but I don't have any special ways of doing that. An easier way is just to measure how much carbon dioxide is created. So there are many ways you can do that. We're going to use just balloons. We're not really caring too much about really precise data on this one. I just want kind of a demonstration that you can see easily, so I'm going to capture it in these balloons. And you can see the balloons, they're all the same type, same brand, even the same color, trying to control for as much as possible here. If you're interested in doing this more quantitatively in a way that you can really count well, you could put maybe a carbon dioxide meter on top if you're lucky enough to have one of those, if your school has one of those. You could also just put on a gas pressure sensor. The higher the pressure inside the container, the more gas must be created inside the container. So therefore you can surmise the higher the pressure, the more active the yeast are. There are many ways you can do this. We're just going to use balloons, though. I like balloons because they're easy and visual. Oh, this one just broke, so I'm going to put a fresh one on. Okay, there we go. So now I'm going to leave them for 24 hours, and we're going to see what happens. Once again, flask number 5 is yeast, flour, and water. 4 is just sugar and water, but no yeast. 3 is the yeast, the sugar, the water, and it's been boiled. And 2 is yeast, sugar, and water. And 1 is just yeast and water. So those are my 5 different treatments here. We're going to check in on them in 24 hours. I'd like you to make your hypotheses now. Make sure you have a hypothesis for each of the 5 treatments. Alright, it's been 24 hours. Let's take a look at our solutions. You can see all 5 lined up here. Let's start with the yeast in the water. The balloon looks kind of like it did when we left it. I don't see any bubbles. Looks like no fermentation happened. The yeast, the water, and the sugar we can see is nice and foamy in the flask, and the balloon is full of gas, presumably CO2. The one that's been boiled has the same stuff in it, but it's been boiled, and that one failed to produce any CO2, failed to ferment. The one that doesn't have any yeast in it also failed to ferment. We see the balloon's totally limp there. That shouldn't surprise us. And the flask on the right, the yeast and the flour in the water, that one did ferment. We can see it's kind of bubbly and it's got a nice full balloon, but it's not quite as full as the other one. So here are our results.